In this video we will discuss about one of the candidates that they have checked whether it is working better than pdcd1, which is the previously tested genes, and also they checked it for a gold standard NFAT promoter or tumor specific transgene expressions. However, they have not mentioned the reason why they have selected NR4A2, not the other candidates in their main manuscript. So let us discuss the results that they have presented. First, they check the expressions of this NR4A2 and compare it with this pdcd1, and we can see that these NR4A2 expressions are more specific compared to the this pdcd1 GFP expressions, as we can see these non-specific results also, and that is even without the activations of the T cells. Then they checked it in different cancer cells conditions, different types of cancer cell lines. First they check it for ovarian cancers, now they check it for a different kind of cancer cell lines. So in the panel A they check it for normal 83 cells, parentals, and the ovarian or the cell lines or cells that are affected with the tumors. So even in the parental 83 cells we can see the non-specific expressions. Thereafter they check it for the different cell lines, that is E0771, MC38, 83, and also we check it with activations of these CAR-T cells using anti-CD3 CD28, and we can see that in all the cases there is a high expression of this GFP compared to the non-stimulated conditions and compared to the parental conditions. In the ovarian conditions we can see the more GFP expressions for both the cases, and the difference is we can see that even in the base cases the signals for the NR4A2s are lower compared to the PD1 GFP expressions. So it shows that the specificity of this NR4A2 promoter for tumor specific transgene expressions is high compared to the PD1 GFP. Thereafter they check it in the in vivo studies to check whether the CAR-T cells are expressed at non-specific sites or not, or basically the CAR-T cells are expressed. Activated CAR-T cells are or we can see the GFP expressions can be seen in the spleen or tumors, and they can see that for the PD1 they are able to see the GFP expression in the spleen, but for NR4A2 the GFP expression in the spleen is less than 1%, and they are more in the case of these tumors, and these things are quantified in the right hand side of the panel B, where we can see that for the PDGF1 even in the spleen the GFP expressions are higher, but for NR4A2 GFP expressions are almost negligible for the case of spleen, but they are high in the case of tumors. Then they check its efficiencies in terms of tumor suppressions and also the survival percentage. In terms of tumor suppressors, we can see that the efficiencies of this NR4A2 is high. It is able to suppress tumors for longer period of time compared to the case of this tumor suppressions abilities are checked with this IL2 is introduced just after the start codon of IL2 sequence has been CRISPR knock-in into the start codon of this NR4 after the downstream of the start codon of NR4A2, and we can see that this interleukin 2 expressions can cause suppression of the tumors for longer period of time, and also the survival probabilities are increased compared to the case where NR4A2 has been knocked down. Thereafter, they also check its side effect in terms of the body fat. They do not see any side effects for NR4A2, neither for NR4A2 knockdown or NR4A2 club with interleukin 12. So once predicting that their NR4A2 promoters has better performance compared to the PDC or this PD1 GFP expression, of this pdcd1 gene. Thereafter they check its efficiencies with the gold standards, that is NFAT promoter. So in that case, what will happen is that the plan is shown in the panel F. Here we can see that this endogenous NR4A2 promoter. So what it will does is the when the transgenes expressions is controlled by this on and off mechanism of endogenous gene, or that is, it is activated when the cells are CAR T cells are activated, and it is suppressed when the CAR T cells are not activated. And for NFAT promoters, is controlled by single transcription factors, that is NFAT. NFAT is generally transduced when the cells are activated, and this NFAT is been or the promoter that controls this NFAT will move into the nucleus and leads to the translation of this NFAT promoter that will cause these GFP expressions formations. So first they check the specificity at different sites, so and compare it for the NFAT and the NR4A2 gene. And for NFAT, you can refer to the paper that we have discussed, how the NFAT promoters they have designed. So first they check the specificity, and they are able to see that for the NR4A2, the non-specific expressions at other parts of the or at other organs of the mouse is low compared to the case of NFAT. And these things they have quantified in the representative image are shown in the panel H. The quantifications are shown in this panel GI. So here we can see that when the cells are activated, non-stimulated conditions, the NFAT has some signals, but when the cells are activated, the signals are much higher for the NFAT, and also signals are higher for NR4A2 compared to the background. And when they use these anti-CAR antigens, the similar results they have seen. And so here the comparisons is not between the non-stimulated, but comparison between MOC, NR4A2, GFP expression, and the NFAT GFP expressions. So in all the cases we can see that GFP expressions are statistically significant compared to the MOC for both NR4A2 and NFAT, but they are higher for NFAT. And when they check it for other different parts of the body, the quantifications of the panel H is shown in the panel I. We can see that the signals are statistically significant for both NFAT and NR4A2. But when they check it for the in vivo studies in the E0771 HER2 mice, in that case they can see that this NR4A2 with IL-2 perform better. And it also has the high percentage survival results. And when they did the similar studies on the other cells, that is MC38HR2, they see the similar results, indicating that the functionality of or the efficiencies of this NR4A2 promoter is much higher compared to the pdcd1. Results for the NFAT is not shown in this main figure, but I think the results for NFAT might be in the supplemental figure. I am not 100% sure. You can refer to the paper in order to understand the details about the experiments, and its descriptions or link for the reference paper is provided in the descriptions. That's all for this video. Thank you for your time.